Today we have a PhD thesis defense by Pavel Sulimov. He is here. He defends thesis titled "Learning Generative Probabilistic Models for Mass Spectrometry Data Identification." The major for this thesis is computer science. He did his work in High School of Economics, and his advisor is Attila Kertesz-Farkas. Am I correct?
Yes, perfect.
He's professor at this High School of Economics, and his advisor, Pavel's advisor. And we have committee consisting of 5 members, myself, Mike Gorshkov from Federal Research Center for Chemical Physics, Professor Andreas Mund from University of Copenhagen, correct? Luca Oneto from University of Genoa, Professor Jacek Perez-Riverol from European University of Bioinformatics, and he's not here right now, but will be later, Professor Sándor Ponger from Peter Pázmány Catholic University from Hungary. And first, I have to make a legal statement. All documents, according to our records, which Pavel submitted, correspond to all requirements for obtaining PhD thesis for this defense. And now let me transfer the word to Pavel, and he will start his talk. Please.
Thank you. Yes, I'm ready. So now let me please share my screen so that you can see it. I guess now it's visible. Yes?
Yes.
Okay, yes, perfect. Thank you. Yeah, just not to see it. All right. Then if it works, we can start. Dear colleagues, dear committee, and everybody who came to this defense, good afternoon. My name is Pavel Sulimov, and today I'm going to present you the results of my PhD thesis. That is topic of learning generative probabilistic models for mass spectrometry data identification. My supervisor is Dr. Attila Kertesz-Farkas. So let's proceed first to the field of my research, and this stays particularly for spectrum identification. So we do not go to the biological data preparation, we do not go to converting of the results from mass spectra to the computer vision, and we just take already prepared spectrum and provide the annotations so that we do not even proceed with protein assembly. So then, what is the basic idea and what is the problem? That when we obtain experimental spectrum, we don't know in advance what theoretical spectrum corresponds to it. So there is no a priori knowledge. So for the reason of figuring it out, we simply take our experimental spectrum, discretize it as well as the theoretical spectrum, and getting real valued vector from experimental spectrum and binary vector from theoretical spectrum, we, for example, can make simple inner product, and this would be the kind of the score that we obtain and that we can trust in terms of how well these two vectors match. And from the biological theory, just the distance between these two peaks, between peaks for Bu and Y ions correspond to the mass of amino acids. But then somebody can ask, what's the problem if it is so easy? Unfortunately not. Because the identification process, it could be hampered by, for example, unexpected fragmentation peaks, then expected peaks, but those that were not observed, or modifications in data, and in fact, correct annotations might be just not included to the reference database. So that all leads to the point that 60 to 70% of the data might not be annotated with high confidence. So then what to do? At first, we start with improvement of the scoring method, and we introduce our method that called Boltzmann. So what we basically do is improve the idea of the scoring that I have shown before, but here, instead of just making simple inner product, we naturally generalize it to the stuff that is presented here with getting this weight matrix W. So we perform the PSM score here, and as well we provide normalization factors. So in fact, it is just absolutely natural generalization of X-score method. And the question would be then how to train this particular matrix W, because other parameters, they are known. So for this reason, we provide training via restricted Boltzmann machines with the following update rule that I provide here. And what is important to understand that this particular learning objective that is state there gives more stable training comparing to other tested objective functions that we have done during all our research period. However, training of RBMs is notoriously hard because originally there are included latent variables, and more to say, Markov chain Monte Carlo sampling is required in order to provide the normalization factor Z. So in order to somehow mitigate the effects of these hampering factors, we have introduced several heuristics. So just one by one. The first heuristic stays for restriction to only observed spectra S prime and to only to possible theoretical spectra H prime. So moreover, we define the probability of match equal to 0 whenever the precursor masses of the spectra are not equal up to some instrument-specific tolerance. So this is general heuristic. Heuristic that would be applied to the first part of the equation that I have shown, to such called positive phase, would be that we suppose H prime to be approximated by the best matching theoretical spectra H star found by X-score method, the method that I have already introduced, that I have already mentioned, that we are generalizing it in fact. And the point is that we are taking not all the matches, but just top matches with Q value less than 0.005. And this leads to a self-supervision approach, and this gives us ability to eliminate of latent variables. In order to somehow make more simple the second part, we take for every experimental spectrum as prime only those H primes that are from candidate theoretical peptides from particular peptide database that we take. And it is very biological and natural because in general restricted Boltzmann machine, we need to go through all the possible stages, I mean, in terms of providing this normalization. But there, for example, we can encounter such vectors like only zeros, only ones, or for example, vector 0, 1, 0, 1, 0, 1, that are biological absolutely not plausible. So that's why we introduced these heuristics. And last but not the least is introducing the heuristics additionally to our log likelihood optimization. So we introduce regularization. It comes from the idea that we have observed in, for example, Humvar dataset, peaks in more than 97% of spectra around 230 MZ and 115 MZ. And these peaks possibly we suspect that correspond to single charged and doubly charged TMT labeling residues. So in order to mitigate the effect of these ubiquitous peaks that appear in most of spectrum, but that absolutely useless for training, we introduce these regularization parameters so that we want the hidden states, the transformed states after multiplying S by W to be different for different spectra observed. And what we simply do, we add this regularization to log likelihood optimization. These are the just results of Boltzmann work, and here you can see the evaluation via target decoy search with plot of FDR curve. Here it is visible that Boltzmann with calibration method that I would introduce later, and we've percolated that they both outperform, for example, PROSIT method, that is deep learning based approach that was published recently in Nature Methods, and that it is outperformed at the 0.1% FDR level, introduces that Boltzmann is more discriminative method. So I mean that it makes nice diversification between the correct and incorrect annotations. And what is also important, that Boltzmann achieved a new state-of-the-art level according to these results. The more interesting fact is that comparing to all these deep learning black box methods, Boltzmann could be interpreted. And here you can see the weight matrix that is just heat map representation. And there, for example, you can see that A is just a main diagonal that stays for the match between experimental theoretical spectra at some exact position. And then B stays for some off diagonals with like water loss, ammonia loss. Then C stays for different losses of amino acids. And what is interesting that there is this skew diagonal D that corresponds to the doubly charged peaks. And one more interesting picture is about why Boltzmann works. So here in the top, you can see the standard scoring, that's in fact the spectral transformation with X-score. And in the bottom, you can see Boltzmann transformation. And the point is that Boltzmann is able to reconstruct unexpected but unobserved peaks. And as well, it reduces the intensity of unanticipated peaks. So it's visible from this comparison. So in fact, there is one, how to say, drawback that this weak supervision that requires PSM results is in fact a chicken egg problem. And we have tried to somehow get rid of this problem, and we have even replaced this H prime not just with results from X-score database search, but just with filtered experimental spectrum. And even though it has outperformed X-score, it was not able to outperform the result that we obtained with self-supervision. So that's why currently this work is like in progress or on hold, but still outperforming of X-score gives us hope that this method might be improved and still work. So after figuring out that we have a nicely discriminative machine learning method, we have just asked ourselves the question like, is this method biased or not? Because we had a thought about the point that machine learning approaches can introduce machine bias, and then it could be reflected in bias in FDR estimation. And for example, this has happened with DRIP method. The result for proving that DRIP is biased is on the left diagram. So there we have taken just the tail of the PSM scores and have provided man-Whitney test. And then it occurred that DRIP methods still can prefer target decoys to peptides, target peptides to decoys peptides. And Boltzmann method with the same testing for biasness showed that it is unbiased. So as soon as we have now this discriminative unbiased machine learning method, we need to have nice source, and this would be a calibration. So we have introduced Taylor, like the new heuristic score calibration method for scoring functions. And just the question why we need this calibration, why not just to leave the method as it is? Why not to leave score this? Because uncalibrated scores may indicate different match quality for different spectra. For example, as it is here on the left diagram, that for doubly charged and triply charged, the distribution of scores differ. And our result is to see some picture like on the right. And surprisingly, we get it with help of applying Taylor calibration. So this Taylor calibration states for calibrating of PSM scores just by dividing them with top 100 quantile of the empirical spectrum-specific node distributions that were observed during database searching. It is shown there what we're doing. So we're just simply sorting the scores, and then this Q100 would be the score with associated p-value of 0.01 at the tail. And there you can see on both left and right diagrams the examples of how Taylor calibration improves the annotation, just for example, on these data sets. And though Taylor calibration can get comparable results with exact p-values, Taylor runs about 100 times faster. So that's why we claim that it is a universal rapid non-parametric score calibration, and rapid is here one of the main words. So that was the main output of my thesis. It has been formed in 4 articles, 4 main articles. 3 of them are in Q1 journals, and 1 more is in Q3 book series. So that was all about my talk. Thank you very much for your attention, and I would be very happy to answer any of your questions. Thank you. I hope that you have heard me all that.
Yeah, Pavel. I have.
Yes.
Sorry.
Thank you very much for your talk. You've been concise and just right in time. And now we have questions session. First questions from committee members, and then all participating colleagues can also ask questions.
You have one question?
And
yeah.
Please, let's go with questions.
Can I start?
Yes.
Okay. Hi, Pavel. Congratulations for your work. Very impressive. I really liked it. Also from my review, I don't know if you saw it. So congratulations.
Yeah, sure.
First,
thank you.
This is my first statement. The second one is that surely the results are pretty good, as it is possible to see from the publication. But what I would like to know from you is what you think is your most valuable result, and that can give to the community something that can rely with in the future, okay? Mm-hmm. And so this in terms of impact on the research community. And from the other side, what do you think is the most interesting technical results? So I saw that you have applied models that are known in a novel way, but do you think that what you have retrieved, what you have discovered, can be also applied in a more general field, and the technical results can be useful also in other field of application which are related, and so on and so forth? Okay.
Yeah,
Is
sure.
it clear, the question?
Yeah, it's absolutely clear. Thank you very much for your questions. Okay. So at first, maybe about the how to say the main impact how to say on the scientific field. So I would say, to be honest, that I would give this preference to bias in FDR. I mean, because this particular figuring out that bias can occur in machine learning method, and we have provided it for drip method, for example. We have shown it in percolator. So this is the points that machine learning methods need to avoid, and providing these particular in this mass spectrometry field, I think that is important, even more important than producing nice calibration or nice discriminative scoring method. And as concerning some general stuff, some technical interesting stuff, I would say that it would be diversifying regularization, because honestly speaking, the journal, I mean, the article where it is published, it was not related to mass spectrometry at all. So it was about just learning with applying this diversifying regularization. And me personally, there I was making better training of deep belief networks with pre-training restricted Boltzmann machines with diversifying regularization. And then I understood that it could be reflected to the point that I got in these mass spectrometry data sets, that I got this problem that I have these ubiquitous peaks, and I need to provide something that would be producing me like the mitigation of these effects. And this diversifying regularization worked. So that's why I would say that this would be the most interesting technical point.
Regarding the biases, do you think that there are other fields completely unrelated, for example, I don't know, fairness in machine learning or something like that, where this kind of problem appears and this kind of regularization can be applied?
In other fields, that means obviously yes, because these bias, it happens because some of just in any case where we can have like unsupervised learning, like where we have these in our case targets and decoys, and then we understand that in fact the background is not just to find targets instead of decoys, but to find well annotations. So that's why when in training, the idea of preferring target to decoys is used, then the biases just hold the idea of this output. And as far as I said, these diversifying regularization, for instance, it could be applied. So I mean that these as far as I already mentioned, like deep belief neural networks, so they're pre-training with restricted Boltzmann machine. So it has just shown better convergence, faster convergence with applying them. So we have been trying it on CFAR data set, for example, for images. So yeah, in a lot of spheres.
Okay, thank you very much for the answer. And again, congratulations for your PhD. Very good work.
Yeah, maybe I can say something too. Can you hear me?
Yes.
Yes. First of all, yeah, very nice dissertation. I enjoyed reading it, and it's also a relevant question for the field to address. And it's also an emerging field like peptide annotation, peptide prediction. So I want to ask him maybe a little bit higher order question. So you said you focus not on the biological sample preparation, neither on the machine part, more on the downstream bioinformatic pipeline. So if you see the whole MS pipeline as one thing and use what you contributed as a piece of it, so how would you say the other parts, the upstream parts, what would be there the kind of critical points which would then affect your downstream analysis?
So you mean like what parts would influence our part of applying the spectrum identification, yes?
Yes, exactly.
You know, to be honest, I'm not a biologist. I can only guess, and I have written it in Thesis that, for instance, like this score functions property, for example, that scoring method should be universal, does not depending on any experimental protocol that is applied. And in fact, this is what we have done. So I mean that in our data sets, we have, for example, these triptych stuff with homeward data, then in malaria data, we have non-triptych cases, then we have different how to say different types of mass spectrometers. And
yeah, so that's exactly what I'm pointing towards. So if you use different enzymes, different instruments, what would be then the challenges you have to face? Yeah.
Yeah, exactly. So some of these mass spectrometers, they are, how to say, provides, so they introduce different types of problems. Yeah, those that we have just annotated, those that are unexpected peaks, then some miss peaks, then some modifications. And the key stuff is that with method we apply and we're using the self-supervision, all these points are neglected as soon as we provide some self-supervision, and then our method can identify. For example, if we would be talking about, for example, post-translational modification, I guess that they could be, for example, learned by our method if we produce enough amount of them in data that stays for the self-supervision, so with this smaller Q value. So I guess that's our approach more or less robust to all these biological problems that could occur.
Good, thank you.
Thank you.
We have Professor Shandor Ongor joining us. Hello. Okay, more questions from the.
Okay, I have a couple of questions. First of all, congratulations. I think it's a really hot topic. It's really relevant for the biology community to have this kind of algorithms, because the way we identify peptides and finally quantify proteins basically define what actually are the biological claims we can do with an experiment. This is really a crucial and really important topic for biology right now, and especially for proteomics. I have a couple of questions. Also comments on the thesis. The first question, and I'm really glad that in the presentation you mentioned PROSIT, because it's the latest algorithm that does use deep learning to do annotations, spectral annotations. And I would like to know your opinion on detail about the benefits, advantages of your algorithm and your work compared with PROSIT, because also is the most, I would say, the most useful or tool out there right now. That's the first question. Related with that, I would like to know your opinion on the use of spectral annotation algorithms, like your solution on how can improve the detection of single amino acid variants and unexpected post-translational modifications. Because we know that these algorithms are really relevant where we are trying to see or annotate variants at the level of the spectra, and also unexpected PTMs, basically PTMs that the user will not was actually not trying to look for it. That's actually my second question. The comment I really like that in the presentation also you specified that Taylor compared with other algorithms. At the very end of the paper of Taylor, you mentioned that unfortunately Taylor do not perform poorly with some specific algorithms. I would like to know your opinion about which are those algorithms. Why do you think it poorly performed with some of the algorithm or scoring methods and so on? Just to know your opinion. Okay, thank you so much, and congratulations again.
Thank you very much. All right, so then let's go one by one. So the first one was about the PROSIT. In fact, at first, we were not comparing our results with PROSIT. And the point that we have been asked to make this comparison when we have submitted our paper to the bioinformatics. But why initially we were not comparing with it? Because in fact, from my point of view, PROSIT and Boltzmann, they are not competitors. I mean, because PROSIT, it re-evaluates just the theoretical spectra. So I mean that it predicts intensities, and it makes it in terms of obtaining results from max quant, and then so from Andromeda, and then with applying percolator, it's make this reevaluation of matches. So that's why, in fact, Boltzmann could have been compared even with combination with Boltzmann, not just as competitors as we did. So this was the point where we didn't want to fight. We were made to fight, and it like makes an extraction of these features like from PROSIT, and the thing why we didn't do this, why we have not made this improvement of Boltzmann with help of PROSIT, is that when this thesis was written, I don't know how it is now, but in February of this year, PROSIT was taking results only from max quant, so it was not taking results from arbitrary database search methods. So that's why if one day the laboratory providing PROSIT would make these improvements, I would be very happy to make PROSIT work both with Boltzmann, and maybe we would provide new state of the art. Who knows? Who knows? Yeah, so this is my opinion about PROSIT. Sorry, I have already forgotten your second question.
The second question is, how do you think the algorithm that you are proposing can be used for unexpected PTMs on the identification and also single amino acid variant validation? If you have seen that your results also can help the user at the very end to annotate those peaks.
Yeah, so this was the fact that I have already mentioned, I guess, that PTMs, this is some specific part, and in fact, when training, when you're talking about unexpected PTMs, if we include more or less feasible amounts of the spectrum, so that were for some reason annotated with really high confidence, so with smoky value, yeah, like with less than 0.5%, then for sure. So it means that it would, I guess, so it would require more research in terms of understanding how to provide, maybe to find some method that already puts these annotations with PTMs in the top, and then to use them in order to provide self-supervision. However, if I would be able to somehow generalize it without making self-supervision, but to make it fully unobserved, then I guess that PTMs might be annotated well as well. So this is my opinion about this. And sorry, your third question.
And the last question is, what are the scoring system or the nature of the scoring system or that are really not compatible, let's say, in some way with Taylor? I think this is what you mentioned in the last sentence of the Taylor paper. I don't know if this is still the case, but I was actually surprised. I don't know if this is the nature of the scoring system or not. Yeah.
Yeah. So I got your question. So the point is that this Taylor technique, it is based on our assumption that there is a correlation between mean and standard deviation. So we have provided it in this Taylor article, and then based on this approach, we were able to somehow compete with exact p-values evaluation. However, on these, in these cases where Taylor doesn't work, this correlation of mean and standard deviation is not that much sufficient. So that's why Taylor is not able to show the same performance.
Okay. But do you have any, for example, do you have any clue if this is happening with MSGF Plus or any search engine specific, or this is kind of generic of, or specific to data sets?
Yeah. We suppose that it is specific to data set, that it happens because of what we have previously discussed, these problems coming from mass spectrometers.
Okay.
Yeah.
Okay. Thank you.
Thank you.
More questions, please? I have some questions.
Sure.
You're talking about PSM level of annotation, right?
Yes.
And I see, for example, if you look at the figure 6 in your thesis, the gain in PSM annotations is so huge compared with other scoring functions like extendem or MSGF plus. What do you think the reason for this such a huge gain, which is shown in figure 6? And also, do you expect the similar gain if you switch, if we go to the protein level identifications?
Okay, so the point of this gain, because where we have this huge gain, it stays for only these baseline methods, yeah? So like this basic X-score, and then this X tandem that uses the idea of figuring out these sequences of B and Y ions with applying this inner product with factorials. Yeah, so but our method, as it applies training of this weight matrix, it makes new some insights. Yeah, so that's why this slide, from my point, it is important. So in fact, in previous versions, we have been showing the slide where we have like matrix with inner products. Then we have the next matrix that, for example, stays for X score with manually calibrated off diagonals, where they had like off diagonals. Then these flanking peaks diagnosed with weight of 50, then amino water losses with 25 and whatever, yeah? But in fact, these machine learning methods, they help to find out these new insights in the data, and that's why we have this gain. So if we're going to the protein level, I don't know, to be honest, how much it would influence. So I would think that, I would say that it would just make the protein ID more confident statistically, and that's it.
Okay, what I was wondering just, what is the source of those spectra? If you identify 50% of spectra, for example, in some experiment, how can we gain twofold or even threefold in PSM annotations?
This is just because of better discrimination. So this is just the idea that I was talking, like it is better discrimination between correct and incorrect annotations, and that's it.
Which means some of the PSMs identified by other scoring function are not
that discriminative. Are not that discriminative, and that's it. I guess this is my idea.
Okay. More questions? We have guests. Maybe they have some questions. So if somebody has more questions, please ask. Okay.
Maybe I want to ask something. So we talk about spectrum annotation tools that you have developed, but how does it look like with MS spectra prediction tools? So what do you know about this, and how do you see there the advantage or disadvantage applying spectra prediction tools?
Spectra prediction.
Yes.
So you mean like
how to compare it to, yes, how to compare it to spectra annotation? What are the pitfalls or the disadvantage and advantage using MS spectra prediction tools like PDeep2 and
like which one?
PDeep2, maybe you've heard of.
PDeep2.
Or do you know anything about spectra prediction tools? Let's say.
No, I wouldn't say that we have been figuring out this field, but in terms of spectrum prediction, I would say that we are making we are making spectrum transformation. Yeah, so that's why I would say that maybe we are somehow related with application of our weight metrics and then providing the new denoise spectra, but I wouldn't say that I have heard about those that you are mentioning. Sorry.
Okay, let's move forward. Now, please, Attila, can you give a talk about defendant?
Yes.
Pavel, please stop the demonstration of the screen.
Of course. Just a second. That's
okay.
it.
Please proceed.
A moment, right. Okay, so this is my time to give the supervisor's evaluation about Pavel. And so I've been Pavel's supervisor for around 4 years, if I remember well. And what Pavel's topic, I think Pavel's topic is innovative, is that it takes the whole peptide or the spectrum annotation closer to this end-to-end type of learning, in which some of the parameters in the scoring function, which have been usually manually tuned, they can be now learned with some machine learning methods. And the weights of the parameters or the values of the parameters can be calculated purely from the given data. I think this is the biggest achievement of Pavel's thesis. I also would emphasize that Pavel's method is fully interpretable. We were considering fully deep models which are not interpretable, but we decided to make our results more like interpretable. And we didn't try very deep models. I think also we've pointed out that these automatic scoring methods can involve machine bias, this machine bias phenomenon. And we also draw the attention of this community to avoid these pitfalls of learning scoring function. In fact, I just mentioned that we also analyzed other deep learning methods. For example, the PROCIT, we showed that it still has some bias in its scoring function, although that bias is conservative. It results in a conservative FDR estimation. So during the work, or during this time while I was working with Pavel, I think Pavel has matured to be a good and young researcher. He has learned all the necessary things about machine learning and the general things of machine learning and statistics and these generative methods. And Pavel also has become familiar with all the scoring functions and the standard methods and softwares and programs and pipelines related to mass spectrometry. So his expertise in this interdisciplinary area. Finally, I would like to mention that Pavel's thesis fulfills all the requirements for this PhD degree in computer science at our university. Okay, my closing remark is that I fully support Pavel's PhD, awarding a PhD degree for Pavel. Thank you very much.
Thank you.
Thank you. Okay, the next step in our meeting is brief talks from committee members regarding thesis. And we can start with myself. And I should say that the theses are related to the area which is called computational proteomics, which is a very important area of the step in the proteome analysis workflow. And from the very beginning, the data processing was one of the most crucial part of identification, because in typical proteomic experiment, we are dealing with hundreds, tens of thousands of spectra, which cannot be manually examined, and we need some tools and that automatically process those data and extract the relevant, biologically relevant information about the sample from those data. And these tools, and most of them, these tools are still suffering from few problems. First of all, computational speed, if we want to increase specificity and sensitivity of the search, and also from biases, which are described in thesis. So Pavel's work was focused to deal with those problems, to increase universality, computational speed, and overcome the biases of existing scoring functions. And from this viewpoint, I think he achieved very nice results in his work. I actually don't have any serious remarks regarding the thesis itself. Just there are a few roughnesses in introductory when he's trying to describe mass spectrometry, but of course they are not crucial for the essence of the work. So the theses are generally well written, in my opinion, and the work obviously meet all standards required for awarding PhD degree to Pavel Sulimov in computer science. Thank you very much.
Thank you.
Okay. Any of the committee member can say about the review, please. Andreas?
Yeah, so I can also piggyback on your comments. I think it's also a generally well-written thesis. I think, yeah, as you mentioned, the beginning, maybe it could have been a little bit more mass spectrometry-based introduction before going into the hardcore bioinformatic part. Could have been a little bit more elaborated, but besides that, the thesis is highly relevant. Also, particular the different softwares which are out there or algorithms which are out there from like ProSets and estimated their bias in terms of their scoring functions, and also systematically evaluate different algorithms in terms of using it for peptide annotation and their correctness and their pitfalls. I think it's extremely valuable, and it was very well done and also very nicely described, and finally also documented in 3 great journal articles. And I think for these reasons, I think I can also only recommend this thesis. Suggest to get the PhD title for the achievements of this thesis. And yeah, so I enjoyed reading the thesis.
Thank you.
Okay, thank you very much. By protocols, we have to ask any of committee member to say a few words about this. So, Luca, please, your
yes.
words.
Okay, so
Shandor. Okay.
Yes, I'm Shandor Pongor, and in fact, I am a user of mass spectrometry. Perhaps this is why I am called user here. And actually, this is probably due to the computer I use. So basically, I like the thesis a lot because it attacks a very important problem, and it does it with a very focused and well-designed way. So I think that both the methods and their evaluations are exemplary in many senses. So I think that this is a good thesis written by a promising young researcher. I found that there are 4 major contributions: the Boltzmann method, diversifying regularization, then the part on the bias of the scoring methods, which I liked a lot, and finally the Taylor method, which is a fast heuristic. So I think these are important contributions, and it certainly tell me that I should recommend this thesis for acceptance for the fulfillment of the PhD criteria. And I would like to congratulate the candidate. Also, if I need to mention just one weakness, but this is also my bias, so I'm still biased, you know. So, and is that the description of the mass spectrometry background could be more detailed, really. But I certainly, so I may not need this, but the general reader would be better served by a more detailed introduction. This is, I think, section 2 of the introduction. So, congratulations again, and thank you.
Thank you very much.
Okay, thank you very much. And please, yes, your few words.
I would say more or less the same thing that I said at the very beginning. This is a really important topic. Also, I would say it's difficult because it's a really hot topic, meaning that a lot of algorithms and tools are under development and coming every year. Then it's really important that the topic of the thesis. I would really like to highlight two things. The first thing, and I put it clearly in my document, is like, this is not only a theoretical thesis, but also a practical and implementation is out there. And actually, the author also take the all the work of making compatible these tools with other existing tools, because sometimes you go out there and then you see a thesis and then you see that there is a lot of a theory about how something should work, but nothing like a practical tool that you can use. And this is really relevant. You can now download these tools and then use it in combination with existing search engines, existing tools like Percolator, and it's even integrated in Crux, which is really relevant. And research can start using the output of these tests straight away, which is actually really an important thing. With that, also, I would say that the author not only take care of improving PSM identifications, but also performance, which is mainly the data that is coming out there and the algorithms that are coming out there not always take care of both variables. A lot of people, there is a lot of algorithms out there that they take care of improving the results, the number of PSM, the identified proteins, but not the performance. And we have seen that in these tests, both things are coupled together. Then the identifications are improved, the accuracy of the identification are improved, more PSM, more protein, but also the performance, okay, which is actually really relevant. I will, of course, recommend the thesis to be accepted for the PhD. I will sincerely congratulate both the candidates and also the supervisor also for the incredible work, and thank you. Yeah.
Thank you very much.
Thank you very much, and please, Professor Neto, can you say something?
I totally agree with what's stated by the previous examiner, in the sense that I really like the work, and obviously the scientific output explains by itself the quality of the work. I fully agree with the fact that there are multiple contributions inside this in PhD thesis. There are practical results, so the solution of a quite important and a novel problem. There is the development of new and adaptation of state-of-the-art techniques, the proposal of a new regularizer that for sure is quite important results, and its implication also in other fields will be for sure investigated and important for the community. And for this reason, I fully support the degree of Pavel.
Thank you.
Okay, thank you very much. Now, Pavel, it's your turn to answer some questions or comments from the review committee.
Okay, thank you. Yeah, I think that mostly those questions that were like direct one, they were answered before this session. Like the only thing I want to add, yes, I totally agree that the mass spectrometry section could have been more humble, but I understand at the same time that being not biologist, maybe I was not able to provide more specificity that was expected from me. Still, thank you very much for your comments.
Okay. Next, by protocol, we have to open some public discussion regarding this. And let me start. I'm thinking, Pavel, what do you think for the next? Are you planning to promote somehow this Boltzmann to the level of ready for use search engine for having interface or something like this?
Sure, exactly. So about promoting, so Boltzmann is already open source available. So it is in Attila's GitHub, and yesterday I have even put a star to him. So I mean that now somebody is following this method. And generally speaking, so it would be obviously fine to. I mean currently, what could have been done that it consumes the MZML and FASTA as experimental spectrum and theoretical, and people can already use it because we have a nicely written way of how to do it, and the key advantage is that it could be done just in one line via Python. So just with specifying the paths to files. In terms of just specifying it once again, in terms of improving the Boltzmann itself, it would be a real, how to say, a big deal for me to make it work in unsupervised way, because still I guess that it could somehow provide maybe some additional heuristics that are based on de novo or whatever point. So I mean, to get rid of this self-supervision as much as possible and to make it like absolutely unsupervised. But this is just my dream.
Okay. What about seeing this Boltzmann in protein discovery among the other tools?
Oh, of course, it would be fine for me. That then I understand that it need be somehow solved in terms of like making a great PR about that, because currently I see that still guys are using some basic methods that are somehow implemented into some tools with interface. So, I mean, because currently I was, for example, looking for the articles that are working about this COVID topic. And now what guys are trying to do is to take these COVID ill people and to find some proteins that are just common for these people. And what I have seen, they are using these methods that are with a nice interface. Unfortunately, this is the drawback from our point. We don't have well-written interface for this. Yeah, what I have said that it could be run currently only from Python command line, but maybe during, I mean, in my next research, I think that it would be important because, you know, I guess there are a lot of scientific results that we even don't know about because they are not that user-friendly, but that have nice, could have had nice implementation.
Yes, correct. Indeed. From our experience as well. You are writing something for bioinformaticians only.
Yeah, exactly.
Okay. Anyone can say something? Well, then we have to proceed with the most important part of defense, voting by committee members.
Yes, yes.
Now, Sergey, it's your turn.
Yes. Thank you, Mikhail. Just now, I will invite all and only members of the committee to the special online room for the voting. I must demonstrate the results of the voting to all the participants of our meeting.
Good.
Yes, that is.
Mm-hmm. Yes. Now, the last words from Pavel.
All right. Perfect. Thank you very much for giving such an opportunity. And first, thank you very much for everybody who came today. Thank you very much for to committee for at first nice voting. I really liked it, and of course for your useful comments, for nice questions, because some of them really make sense in terms of what to do next and what the problems were and what disadvantages. So thank you for covering all this stuff as well. Thanks to Sergey Sivantev for organizing all this stuff, because currently I'm located not in Russia and it would be a problem for me to come to university. So thankfully to coronavirus, in these terms, we can do it online. Yeah. However, as well, I want to, of course, at first say many thanks to Attila. Attila, thank you very much. Yeah, because we know each other for already 4 years, and through all these 4 years, you have been always providing me with your, like, sharing your invaluable knowledge, like giving more ideas, and you were just making me provide even more ideas as well, and getting even more knowledge. So it was not kind of competition, but it was vice versa. It was the point of getting more and more. And as well, I'm really happy with Attila. We have been doing it like a pure science. I mean, that we have been not conquering for any grants, for any sponsorship. So we have been doing just a pure science, and I'm really happy about this, because these results, they are just coming from my heart. So this was done because I wanted to do this. And as well, Attila, thank you for, I mean, Attila was the first man who was responding to my emails the same day that I was writing it. And I guess a lot of Russian people suffer from this problem. And yeah, thank you very much. So it was nice international experience, and I really liked it. Thank you very much. And finishing the official thanks, I want to thank a lot Russian government for making long winter holidays, because in Russia from 1st till 9th January, there is New Year holidays, and it was perfect opportunity to work on my thesis. So main results I have obtained during these holidays in my 3 winters that I have been working with Attila. And as well, if coming to personals, I want to say many thanks to my parents, grandparents, to my family, because through all my life they have been pushing me to do science. They were supporting my wish to do science, and I understand that the result that today I'm here, and these positive votings, it is fully thankful to them. Spasiba. Spasiba. And as well, thanks for main thanks to my lovely wife, because when I was coding during the night, she didn't say a word during these 4 years. So thanks a lot to her. And what I want to say in conclusion, that for all my life, I have been doing science as a hobby. I mean, through all these 4 years, I had main work, and the science that I have been doing, I was doing it during evenings, nights, and early mornings. And with now obtaining these positive votings on the results that I have obtained, I truly believe that finally, perhaps, my hobby would become my job. Thank you all.
Okay, thank you very much. At least you find something to thank the Russian government for.
Yeah, exactly.
It was difficult. Okay, thank you very much for everyone who participated in this thesis. And we congratulate Pavel, both Pavel and his supervisor, Attila.
Thank you.
Thank you. Thank you.
For this work and for the successful defense. So thank you very much.